Morning, everyone. So we probably can start today's seminar. So it's my great pleasure to introduce today's speaker. I try to speak his name. So Dr. Kaustubha Qanungo. I'm not sure pronunciation is good enough, but I try. So, but in our department, we call him Kaus. So it's much easier for me to speak. So Dr. Qanungo received his bachelor degree and master's degree in India. In 1995, he received his PhD from Indian Institute of Technology. So he always studied the virus. In 2001, he received a postdoc fellowship as the Department of Molecular Biology of the Cleveland Clinic Foundation in the United States. And he published a bunch of papers about virus. He studied the virus of stomatitis virus. So, and this is a negative strand RNA virus. This data has been published in PNS journal. It's a very prestigious journal in scientific research field. After that, he moved to the Case Western University for 6 years, served as a research associate. In 2011, he moved to Charleston and served as a full-time faculty at the Trident Technical College. And we are very fortunate to have him in 2019. He moved to Clemson as a lecturer. Privately involved in teaching virology, but also he did very good outreach work to disseminate information about biological sciences to general public. So today he will give a very interesting topic. I think now everyone suffer from the coronavirus and the very bad pandemic and in the history. So his talk about coronavirus, the untold story, a molecular insight of the replication, pathogenesis, and vaccine development. Kaus, please.
All right. Thank you, Jicheng, for the gracious introduction. I really appreciate it. And good morning, everyone. So as we know that I'm just named the slide, Coronavirus, the Untold Story, but for many, this story has been told many times. I just put it in the slide or in the topic so that I can just attract more people. I guess that was the purpose. But some of the information could be new to if you're not in the field, but we'll see that how it unfolds. But it will just give you a comprehensive idea about the virus. And usually we talk about the pathology as well as the transmissibility, the epidemiology, but we are going to talk about a little bit detail about the replication strategies, the pathogenesis, and some of the insights of that, which could be some interesting information about that. And it is almost impossible to follow the coronavirus, even being a virologist, because there is an explosion of information every day. And if you even just keep 1 or 2 hours for searching literature and keeping your update, it is almost mind-boggling to keep yourself updated. So before I start, I just like to give this idea that if you're just really interested in virology, and many of you already know this sites, I just highly suggest to look at this. It's called, there's a podcast known as This Week in Virology, and this was started by Professor Vincent Recaniello a long time back. And usually it's used to be weekly. Now it is bi-weekly, every Thursday and Sundays. It's almost 2 hours long, but it's every way it's worth it to keep yourself updated. And when you're working out or anything like that, you can just learn a lot of stuff from there. And this is a very good resource, and I highly recommend yourself that you may not find many answers from this talk, but if you just look there, you may find. And if you just go to their website, you are also going to find some of the specific show notes, which gives you a bigger perspective to look up something specific directly from primary literature. All right. Well, with that, I will just start that the outline of the talk that we are going to talk about a lot of things, and we'll see that how it goes. It could be a little bit information heavy, and I apologize for that. So we're going to talk about a classification, evolution, then structure, really not in detail, just a recap and overview of the infectious cycle. And we are going to talk about a little bit more detail about the entry in the host cell. And there could be some new information that you may find. And here, this genome transcription, replication, and translation is probably one of the area that that could be very new to many of you, which is a remarkable process by these coronaviruses that we are going to see. And then we are going to see that how the coronaviruses basically assemble or replicate, transcribe inside the cell, and some of the clever, cunning strategies to evade the immune interaction by the cell, and looking at the pathogenesis, and finally the antiviral vaccines. So we'll just touch that. So with that, we can start with where they belong, and the coronavirus family is a large viruses, and their genome size are in between 28 kilobase to up to 35 kilobase, and these are the largest RNA viruses that is out there. And you can see that there are many of these. This orthocoronavirus is a subfamily, and particularly the viruses which we are looking at, that these alpha coronaviruses, there are two strains, these HCOV 229E and NL63. And from beta coronaviruses, this HCOV HQ1 and HCOV OC, these and 43. These are all circulating coronaviruses which can cause about 15% of common cold to the human being. So these were already known. And then you can see that there are some other, which is the SARS-CoV. Then this was the 2002-3, the epidemic that broke out and it was contained. Then the MERS, which is the Middle Eastern Respiratory Syndrome, is also, this was 2012. And then there are still, there's a little bit of sporadic cases still today. And then the SARS-CoV-2, these are all beta coronaviruses. And then if you just look at the ancestry of these beta coronaviruses, you can see that they can infect a variety of mammals. And and from those mammals, if you can see that the ancestors are usually either bats or avian. Now, the avian lineage don't infect the human being. The avian lineage are the gamma and the delta coronaviruses, and they are not infected by the human. But the human coronaviruses, as well as many mammalian coronaviruses, they do find their ancestry mainly in bats. So that's basically the ancestry. Now, if we just look at that the zoonotic reservoir in the bats, at least 500 or different types of coronaviruses has been sampled, and there could be more than even 5,000. These are heavily undersampled, and that is the main reservoir. And what happens that sometimes these spill over to some intermediate hosts. Say, for example, the classic SARS was a palm civet cats, and then for the MERS, it was the dromedary camels. And from that, they came to the human being. Now, for the SARS-CoV-2, although I put a picture of the pangolin, we still do not know that what is the intermediate host. And one of the way that the SARS was probably contained very quickly, the 2002-3, that as soon as it was known that these civet cats were are the intermediate host, they were all basically slaughtered and taken care of, and it basically contained in that way. Another interesting thing that we are going to see, that the SARS-CoV and the SARS-CoV-2, both of them use the ACE2 receptors that you know very well, whereas the MERS actually use a different receptor, which is a dipeptidyl peptidase 4 different receptor. Just another idea is the MERS is much more lethal compared to that. And you can see that there was about 2,500 cases, 881 death, and the lethality of this is much, much more. So going into the next area that we are going to look at, that how these RNA viruses evolve and what we mean by these variants and what's going on in that, to understand that this is just a basic virology perspective that we are looking at. So what we are looking at, that right here, x-axis is the genome size, and you can see that this is about coronaviruses. You can see 10 to the power 4 to 10 to the power 5 nucleotide is about right here in the middle. This is a coronavirus genome size. And if we look at all the different viruses, remember that the coronavirus is a plus-strand RNA virus, and the mutation rate is pretty low. And you can see that there are some of the coronaviruses, some of the other plus-stranded RNA viruses may have very high mutation rate. And we are going to see that why this mutation rate is so low for the coronaviruses, despite being such a large genome. That's number one. But one thing you have to understand, that even at this level, the coronavirus genome, if you compare it with a bacteria, you can easily imagine the number of folds of mutation rate in viral genomes. So our mammalian system does have extremely robust proofreading activity of the DNA polymerases, and it does the proofreading ability very well. The bacteria does have a primitive proofreading activity, but there is some. Most of the viruses which use usually the cellular DNA polymerases for their life cycle, and RNA polymerases make less mistake. And that's the reason, if you just look at the single-stranded DNA, double-stranded DNA viruses, you can see that their mutation rates are much lower compared to that. On the other hand, if you just look at the RNA viruses, which do all of them do code their own enzyme, because in the living world, there is no enzyme that can copy an RNA from an RNA. So that's why all the RNA viruses will contain these RNA-dependent RNA polymerases, either in the virion, or they will be coded. Say, for example, plus RNA virus, they will be coded by that. And most of them make lots of errors. And every replication cycle, there will be these mutations that will accumulate in there. And we're going to see that in the coronavirus, other than these, although they make these large mutations, they can maintain this large genome because they do have a proofreading activity associated with the virus. We're going to see that. So what does this mean? So this means that when in an individual, if you take a viral sample, then even in the single individual at simple point of time, if you do a deep sequencing, you will find a mixture of these virus genotypes. So here, each line actually represent a genome, and each of these are the mutations. But when you sequence, usually just by a normal technique, this is the representative genome. We do not get these altogether unless we basically do these deep sequencing out of one sample. So what we mean by this gives a concept which is called the viral quasi-species, that at any time in any animal in the cell, there are many different types of individual virus genome sequences in there. And that means that this is just not a single sequence. The single sequence is just a representative sequence, which may contain actually many of these examples of that. And this gives you an idea that why these variants are basically selected over the period of time. And here I'm just showing you this slide. Let me go here for a second. And this actually was shared with us long time back with Professor Barbara Campbell. Actually sent us an email to look at this site. And it was an amazing, which actually shows that right from the beginning, 2019, December to up to today, that how these strains evolved over the time. So if I just play this, just take a look at how the different, this is the evolution of the virus. And I can just fast forward this a little bit. And you can see that over the period of time, that how these viruses basically have all the different lineages, which these different lineages are marked right here. So what it all means that if you just look at these viruses, this virus, this is the genome sequence, and the representative of the genome, you have this ORF, open reading frame 1, 2, and then we're going to talk about that. And these are mainly the structural proteins. And this S is the spike protein region, and each of these are the mutations that are being seen in there. So you can see that over the period of time, that no matter what, all these viruses evolve, and this is due to the selection pressure. And in the case of an epidemic and the pandemic, when there is a lot of people are getting infected due to the antibody evolution, this will happen all the time. And sometimes, again, this is in the many literature, this is a very alarming thing for many. Many of the virologists do think it is not alarming, and there is a war out there in between the epidemiologist and the virologist that what do you mean by more transmissible or more deadly? They do not like it, and I am not going into that controversy, and you can look it up on your own with this. So here we are looking at that what are the things that we know for sure without going into the controversy. So this is an UK variant. And what is happening in here? Here we are looking at that there are a lot of mutations have been accumulated, that 17, 18 mutations in many of these variants. And these are some of the important ones. A deletion at the 6970, then this asparagine to tyrosine at the 501, and you can see that this P61H, these are some of the key residues, which I was just listening to this Daniel Griffin, who comes into the podcast in the virology, and he was saying that some of the monoclonal antibody therapy, which is the passive immunotherapy, they are not working very well if they have these particular mutations. So this is an outcome of the monoclonal antibody therapy. But this is nothing to do with the viruses, and this is for the mass population. This is not any alarm as such. So here you can see that there are some of the notable mutations in the South African and the Japanese and the Brazil strain. And all you can see that looks like that this N501Y asparagine to the tyrosine, this is present everywhere. And you can see that's E484K, that this is also present anywhere. So what does this mean? That definitely means that as these are now more prevalent these all stem from a specific variety, which we are going to talk about in a second, which is D614 mutation from that lineage. And it just gives that the idea that definitely these viruses do have some kind of fitness compared to the other, and they are selected. Now, the fitness does not mean that they are more lethal, that they are not more transmissible. There are many other factors that can contribute to these attributes, and we have to be very careful using these terms. But more and more data are coming up, and a couple of publications also coming up, and you can just look at them to have a very comprehensive idea about what these really means. But as I said, there is still a lot of controversy in between the epidemiologist and the virologist about the variants as such. Now, if we just look at the structure right here, you all know this structure, and I am not going to describe this structure. Everybody knows that this is a household name. I'm just still going straight into the overview of that. So what we mean by a plus-stranded RNA virus, you have the long RNA, which does have a 5' cap and a 3' poly A tail. And if you just look at the polarity of that RNA, it is a 5' to a 3', and that means that it is a plus strand. If it would have been a 3' to 5', it would have been a negative strand RNA, just like your influenza virus is one of them, like a negative strand RNA viruses. So this looks like the RNA looks like an mRNA, and it is covered by a nucleocapsid protein. And to replicate this, you need a virus-coded RNA-dependent RNA polymerase, RDRP. The reason that there is no cellular enzyme that can copy these RNA into the RNA. But what happens that once the virus goes into the cell, this can be read as an mRNA. And let's see what happens at that step. So once the virus, and it will go into the entry, once the virus enters into the cell and delivers its genome RNA into the cell, it is read by the ribosome, And the ribosome makes these large two polyproteins. One is 1A and another is not really 1B. It's 1A and B together, 1AB, but it's in a different frame. And these polyproteins are cleaved into individual proteins. And these include also the RNA-dependent RNA polymerase, as well as other accessory protein, which set up the viral factory to produce the virion inside these double membrane vesicles. And these double membrane vesicles are also induced by some of these produced viral proteins. And inside these mRNAs are produced. And once these mRNAs are produced, they are transported back into the cytoplasm, where they are translated into the structural proteins of the virus, as well as inside these, the whole big genome is also synthesized, which will be packaged inside the progeny virion. And in the ERGIC complex, which we call the endoplasmic reticulum and Golgi intermediate complex, these viruses are assembled and then they are released. And we will talk about these steps. So the first thing that we all know household name about the spike protein and the ACE2 receptor. So the spike protein is a trimer. So you can see that there's a trimer and there are two subunits, S1 and S2. And it is inserted into the membrane, which is the viral envelope. Now, coronavirus is an enveloped virus. That means it contains an envelope, and this is derived from some kind of cellular membrane, and it is a phospholipid bilayer. So here is your spike protein. And this is, you can see that. And here, this is an overview that what we mean by the S1 and S2 domain. So this RBD is the receptor binding domain. And here we have a very unique site that is what is called a furin cleavage site. This is a new addition of one protease cleavage site that is added to this SARS-CoV-2. And we are going to see that what these means and what is the consequence of that. And to consequence of that. So let's take a look at these structure spike proteins. So if you just can look at the molecular structure of the spike protein, and you can easily see that this the purple areas are conserved, whereas these areas are more variable. And you are looking at the side view of that. And right here is the fusion machinery or the fusion peptide. This is hidden. And this is, you are looking at the top view. So if you just look at that these residues which bind to the ACE2 domain, this could be very, very variable. And depending upon the variation, of course, the kinetics of the binding can change over the period of time. So you can see that these are three monomers that are in there. The interesting part is that if you just look at these particular residues, these are some very conserved residues, and these gives you some of the very signature structure of the spike protein of the SARS-CoV-2 specific spike proteins. And we're going to come over that. So here, if we just look at the structure of the spike protein, you have a signal peptide because it is secreted and it is a membrane insertion in there, and that's cleaved off. And then you have an N-terminal domain and followed by a receptor binding domain. Now, what you have right here, this FP is the fusion peptide and IFP is an internal fusion peptide. And you can see right here that for all coronaviruses in between this S1 and S2 domain, there is a protease cleavage site right here. And there is another cleavage site, which is called the S2 prime cleavage site. This just only one of these cleavage is not enough to activate the fusion peptide for the membrane fusion. This is, both of them is needed. And another thing is that sometimes people say that if this S2 prime cleavage site is not easily accessible by the proteases, unless that S1, S2 cleavage site is cleaved and this leaves this S2 part, and then the S2 prime cleavage can happen. And that ensures that it binds properly to the receptor, and this cleavage of S2 doesn't happen without binding to the ACE2 receptor. And we'll just come to that in a second. Now, what happens for the SARS-CoV-2, this right here, RRAR, is a furin, which is a subtilisin-like protease. This is an extra site furin or furin-like protease cleavage site that is acquired by this. So now what you can have 1, 2, 3. And the scientists are saying that this, or there is already a paper which tells that this furin cleavage site is probably one of the reason that the rate of infection is much more. So the furin cleavage site needs to be cleaved. S1, S2 cleavage site needs to be cleaved as well as those. So it may be that what happens, that accessibility to the S1 or S2 cleavage site, if before that this furin cleavage site is already cleaved, it is probably easier for the proteases to access this site. And these are some of the speculations from this paper. So what is the reason that this cleavage is necessary? So if we just look at this structure, when the virus gets into the body, this receptor binding domain is the one which interacts with the ACE2 and locks the virus onto the surface of the cell. Then what happens, either the furin as well as, the furin or the S1, S2 protease site, they will cleave this part, and then the S2 prime site will be cleaved, and this internal fusion peptide, which is a hydrophobic residue which cannot be exposed, is exposed, and this is inserted into the plasma membrane, which brings the viral envelope to fuse with the plasma membrane. We're going to see that in a second. So here is an interesting way that how the virus enters. So I'm just going onto this side first. So these are the spike protein, binds to the ACE2, and TEMPRESS, which is a transmembrane protease, serine type 2, and this basically cleaves these, either the furin or the S1, S2 site, and then the released hydrophobic end of the peptides is inserted into the membrane, which causes the membrane fusion. And this chemostat is one of the drug that is being developed but with the Denmark University of Aarhus, and they think that this protease this is an inhibitor for this protease. But on the other hand, we all know that why initially hydroxychloroquine was pushed for that. Even a paper was published, which later was retracted, particularly the efficacy of that, is that there are two ways that the virus can enter the cell. This is the virus enters at the plasma membrane right here, which is the TEMPRESS and furin, we really do not know, mediated, but means that the cleaving of the S1, S2 and the furin site is caused by either TEMPRESS or furin. On the other hand, there's an alternative way where an endocytosis of this, and this is also ACE2 mediated. This is a clathrin-coated pit is formed, and an endocytic vesicle will be formed. And inside the endosome, it will be very acidic. And in the acidic condition, this cathepsin L is the protease which will cleave and cause the membrane fusion with the endosomal membrane, delivering the RNA. So this process happens at neutral pH at the plasma membrane. This happens in the acidic pH in the plasma membrane. And the hydroxychloroquine is supposed to block this step. But again, as the virus used the plasma membrane for entry, that's the reason that hydroxychloroquine did not have any function. And we all know that what happened to this paper, even it is a Lancet paper, which is a group from Harvard, and this was retracted after that. So we see these type of, a lot of these kind of data is out there, which is controversial, but I'm not going into that detail of that. So here is also another idea that how the fusion basically happens. You can see right here is that these are the receptor binding domain, and you are looking at the plasma membrane, phospholipid bilayer, viral envelope, which is also derived from some kind of cellular membrane, either ER or the Golgi, but it's a phospholipid bilayer. And this is your spike protein. It's a trimer. F is the right here is the hidden fusion peptide is inside. It is not exposed to the outside because of this hydrophobic part. So the prime is that protease cleavage, which is 1 and 2, that basically primes and opens up the protease cleavage site 2. And remember that this is the furin site and the S1, S2 site. This is the S2 prime site. Once this happens, see that the hidden red area is those fusion peptides that is inserted into the membrane. These are the hydrophobic part. And then this middle portion, these trimers, they form a 6-helix bundle. And you can see this 6-helix bundle, basically, which you call as a hairpin, bring and draw these membranes together to cause a fusion. Now, the virus can deliver its payload, which is the RNA inside the cell. So let's take a look at a great, this is from Janet Iwasa's lab, and a great idea about that. I just made a little bit of notation. So let's take a look at that. So here we are looking at these spike proteins, you all know from the virus, and the nucleocapsid packaged inside, wrapped with the nucleocapsid protein, the RNA. These are the tempress, yellow-orange, and the ACE2 receptors are in purple. So the spike protein comes in and locks, and these are the glycosylated residues that you can see. So there are 3 monomers of the spike protein, and each binds to one ACE receptor, so there will be 3 of them. That locking helps these tempress to access the S1, S2, and the furin cleavage site. So they are removing these monomers, which are the S1. These are removed. Now, the next part, which is not shown, is at this point, the S2 prime cleavage happens, and this is the fusion peptide falls onto the plasma membrane and inserts. As they are hydrophobic, they are inserting into the membrane. And this is the conformation chain which draws these 2 membranes together. And now the payload can be delivered inside the membrane. So this is basically the molecular process of the fusion. All right, now let's look at some of these ideas that we all know this D614G mutation, and already now it's published that this basically caused a little bit more binding affinity of the spike protein to the membrane. And why it is so, what is being shown in this cartoon as well, that this mutation keeps these 3 monomers of the spike protein in an open conformation, and that has a higher affinity than these closed conformation. And we can see that now the global spread is like that. Almost all the circulating strain is based on now this D614G mutation, including the variants. And this paper has been published in Nature as well, that with the structural basis of the higher infectivity of these. All right, so next we are going into the genome transcription, replication, and the translation. So the virus do contain a long chain, you can easily see, and it contains a long open reading frame, which is right here, is 1A, then 1B, and then many of these structural proteins. And you can see that there are many other different accessory and structural proteins in different SARS, just giving just an idea that how diverse these could be. So S is right here, is a spike. Then you have an envelope. Then you have this membrane proteins, and you have this nucleocapsid protein, matrix protein, and nucleocapsid protein. And then these gray ones are not present in all of them. There is also another HE hemagglutinin esterase that is not present in SARS as well. So there is lots of similarities, some dissimilarities as well. But let's take a look at how this virus can produce 27 different proteins from just 10 ORF. So you can see that these are the open reading frame, and it can produce 27 different proteins out of that. So we are going to see that from ORF 1A and 1B, this is produced as a polyprotein, and after a cleavage by a protease, a large number of protein is produced from here. Whereas mainly the three terminal end of the viral genome, Those are mainly the structural proteins of the virus. So let's first take a look at what happens. So once the virus gets into the cell, the genome is delivered. It first produces the 1A protein, polyprotein, and then it produces an 1AB protein, which is a result at this junction by a ribosomal frame shifting. And after that, the RNA polymerase from these is formed. This RNA polymerase is now essential, which is the RNA-dependent RNA polymerase, to do the rest of the process. So then what happens? You have the full length of the plus RNA, and from those, these subgenomic RNAs are produced by these RNA polymerases. So how this happens? The first step is the subgenomic mRNA synthesis. And in the second process, it produced the full-length genome of the virus, and also that can do additional round of translation. So let's take a look that this could be a little bit complicated, and let's see that how this happens. So we are looking at this middle one, which is the plus 5' to 3', this dark green strand, that is coming inside the cell as the viral RNA. That is also being translated into these RNA polymerases. Once these RNA polymerases and all these proteins are produced, the two things happening. The first one is that we are looking at the bottom portion of this. In the bottom portion, what happens that the RNA-dependent RNA polymerase will try to transcribe from the 3' end, and it will try to go towards the 5' end. But it does not go all the way to make the full length of the minus strand intermediate. Instead, what it does, it starts from the 3' end, go to a particular area, which we call as a translational regulatory sequence, and then jump all the way to somewhere in the 5' end and finish the 5' end. And this jump causes all these subgenomic mRNA production. So all of them do contain an exact 3' end start and a 5' end stop. Now what happens, the same RNA polymerase, remember that this olive strand is the intermediate, and this will be joined together. This is the intermediate. So from the plus, you are getting the minus back, and you are getting the plus back again from plus strand RNA. And this is the final product, which is one of these subgenomic mRNAs. On the other hand, the other way what it can do, that we are looking at the top portion now, that from this end, without jumping, it can transcribe all the way up to the 5' end to produce a full-length minus RNA. And that minus RNA will now act as the template to get back the full-length plus RNA. That is the genome of the virus, which will be packaged inside the progenivirions. So one set is the replication of the genome full length, and the other one is a subgenomic mRNA production for the protein production. Now, very interestingly, if you can see that all these subgenomic mRNAs, their start protein is different. See, right here is 2A, then HE, or HE, right here, S spike for all different proteins. These are not SARS-CoV-2. They doesn't have an HE, but just a representative of that. And then they do have the other sequences at the end as well. But very interestingly, what happens from the subgenomic mRNAs, only the first gene or the first region is only translated. So say, for example, this one is the N. So here, the N protein or the nucleocapsid protein is the only one that is going to be translated. From here, the matrix protein is going to be translated. From here, envelope. Here, SA. So here, it's only the spike. None of the other is going to be translated. Only the first one is going to be translated into the protein. So why it does this whole lot of complicated way? It's a very amazing way that how intricate is the molecular mechanism of this transcription and the genome replication. And it gets a little bit more complicated here, a little bit. So here we are looking at this two ORFs, which are rep1A and rep1B. So what happens that during the translation, this polyprotein is produced, and then there is a ribosomal frame shifting. And how it happens, the ribosomal frame shifting, that in this junction of 1A, 1B, there is a slippery sequence, which is this UUUAAC. This is a slippery sequence, which is followed by this. This is right here is the stop codon for the 1A, the stop codon for the 1A. And then there is an extensive secondary structure, which we call as an RNA pseudoknot, which is present just after this stop codon. So what happens as ribosome is moving in here, it slips on this. And during the slipping, it sometimes go out of frame and doesn't read that stop codon as a stop codon, and keeps on moving through this region, which allows another polyprotein synthesis. So now you can see that from these polyproteins, these 16 proteins can be formed. And why the virus is actually doing this, we're going to see that in a second. So here is your RNA pseudoknot. So this is your polyprotein 1A. And from the polyprotein 1A, you can just look at that. These are the proteins which are formed. In SP1, we are going to look at this, which is an interferon antagonist. And there are many different proteins. Your 3, 4, these are produced used for some of the membrane modifications. And then the polyprotein 1AB is responsible for producing most of the RNA-dependent RNA polymerase holoenzymes. And this one also needs NSP7 and NSP8. So NSP12, NSP7, and 8 produce the RNA-dependent RNA polymerase. And another thing is I'm pointing out here, NSP14 is a 3' 5' exonuclease, which is an exo-N, and this is responsible for and this particular enzyme is responsible for the exonuclease activity. And this is one of the reasons that we are going to see that remdesivir does not work very well. So why you have this double mechanism? The reason for this, if you just look at that these are the proteins which are produced in the molar amount of these proteins are different. So this is the reason that all of these proteins are produced twice, and that's why their amount is more, whereas these are produced in less amount. And this ribosomal frame shifting is controlled by this RNA pseudoknot, which makes these protein in a perfect molar amount. And this is basically the structure of the this area. So all these polyproteins, they have these cleavage sites, and these are cleaved by a main protease, which is this NSP5, and this is a 3CL-like proteases. And these proteases basically cleave these protein for these individual proteins. And these are all non-structural proteins. These are produced only in the cell during the virus reproduction, or which is transcription and replication. None of these are the structural proteins. Remember that the structural proteins are all at the 3' end of the genome, which is basically right here. So right here, this part is the structural protein, which will be done later. So in the first step, this is basically setting up the factory for doing everything that is needed for making the virus. And now we are going to take a look at, I'm just for time, just skipping these, that how this happens, that this is the 3' end from where it starts, and how this TRS jumping basically happens. This is the transcription regulatory loop, which is also present towards the 5' end. And how this movement happens, that as the virus is starting to transcribe from the 3' end, it reaches this TRS, or the transcription regulatory region, which is a very similar sequence to this. And this causes the RNA polymerase to jump. And often sterically, the process is like this, that there is a hairpin formation, which causes the RNA polymerase to shift and produce that 5' end leader sequence from that. And this is the way that all of these RNA polymerases start at the 3' end, exact 3' end, and exact 5' leader sequence in this process. All right, so this is the hollow enzyme of the RNA-dependent RNA polymerase. Okay, so then we could just look at the RNA-dependent RNA polymerase, and we're just going to talk about a couple of different things in here. This is basically the double membrane vesicles which are formed inside the cytoplasm, and this is set up by the coronavirus, a couple of different NSP3, 4, and 6 proteins. So what it shows very interestingly, like here, that this is the micro environment in these replication transcription complexes, and see that what kind of proteins are present here: translation proteins, transport, catabolic processes. But see that right here, these are the coronavirus's proteins, and the ones in the red are all the non-structural proteins which are present in these replication transcription complexes. NSP1 is not, which is also a non-structural protein, and these are all the structural proteins for making the virus particle or the virion particle. They are not present inside this. On the other hand, this is the nucleocapsid protein which enwraps the RNA that is present in here as well. So what does that mean? So this basically means that these are some of the cryo-electron tomography of this, that these are the double membrane vesicles inside which these RNAs are produced, and then they are transported through a pore, and this is made by the virus. This is a viral pore from NSP3. Through these pores, these encapsulated RNA, which are the RNA coated with the nucleocapsid protein, goes into the cytoplasm, and either they are going to do this from this. They are either going to do the genome replication to be packaged inside the virus, or they are going to be going as mRNAs, which will be translated into all the structural proteins. And this is how they hide the replicating viruses inside these RTCs. The reason for this is mainly to evade the host immune response. So how it hides the host immune response? Very quickly, that the virus double-stranded or the single-stranded RNAs can be easily detected by the TLRs. But the RTC formation evades this TLR detection because they are sequestered inside those RTCs. But finally, the host catches up, and that causes an acute infection. We talked about this NSP1, which is a virulence factor. This is a very interesting protein, one of the structural proteins. And see that this protein basically does one thing. It binds to the mRNAs or a particular region which doesn't allow the mRNA to enter the ribosome for the host mRNA protein synthesis. And this is the host translation suppression. On the other hand, all the viral mRNAs do contain a long 5' UTR, or the untranslated region. And due to this, when this NSP1 binds in here, it doesn't interfere with the viral transcription or viral translation. But so experimentally, it has been proven that if there is an increased linker is produced, is put into this N-terminal and C-terminal domain of this NSP1, then this actually causes even the suppression of the viral mRNA synthesis. So this is a very clever protein which shuts down the host mRNA response, basically, and that is one of the reasons that you have a delayed interferon response, because none of these proteins are going to produce any mRNAs for the antiviral state of the cell. So these are some of the structures. We're not going to go to the detail of that. All right. So here what we are looking at, that this is what is the problem, that when you have an early IFN signaling or the interferon signaling, usually you will not see a disease trigger. On the other hand, if there is even no interferon signaling, still you have probably a mild disease. But what happens that when you have a delayed IFN response, and this is basically being done by this NSP1 protein, which is shutting down preferentially the host protein synthesis, you have a late response of the IFN signaling, which cause a cytokine storm, because your immune system is basically startled, which causes this high level of the the cytokine release, and that is basically the pathology of the disease. And here we are looking at that how this happens. All these are cytokines like TNF alpha and then IL-6, IL-1 beta, CCL2. These are all the inflammatory mediators, and it can cause these tissue damage extensively in the lung tissues. All right, so after that, we are quickly going into the antiviral strategy. These are some of the areas that where some of the target monoclonal antibodies are working, like Regeneron or the Lilly. And I'm not going into the detail of that, and I just put remdesivir as well, because see that this gives you an idea that why in vitro studies may not corroborate with that what is going on in vivo in a human being as well. So we'll come back to that. So how these antibodies are developed, the monoclonal antibodies, these are all humanized antibodies where you have this transgenic mouse platform where their original mouse antibody genes are basically deleted and grafted with the human antibodies and antibody sequence or for the variable region. And these are basically challenged with the virus to raise these monoclonal antibodies, which are selected. And these humanized antibodies are different than the chimeric antibodies, which does have much less immunogenicity. And these are mainly the cocktails from Regeneron or Lilly, and many other are doing that. And this does have very high efficacy, but an extremely expensive study, extremely expensive to administer, and this will not be a mainstream treatment for COVID-19, though, unfortunately. So here we are looking at that this was a paper, science paper, early 2020, that remdesivir can cause a chain termination in vitro. Great studies. You can see right here the RDRP complex, where here you can see that elongated RNA. You don't see that. So the remdesivir was a nucleoside analog, and you can see it is bound to the RDRP or the RNA-dependent RNA polymerase of the virus. But now you see that the exo-N or the NSP14 removes that the remdesivir from this site. And this is one of the reasons that this is the only virus, particularly the RNA virus, does have this proofreading activity. And that's the reason that none of the nucleoside analog drugs are working very well with this. So that's one of the reasons that this was the WHO's rejected, but still it is some marginal improvement with the severity of the disease, and still it is a clinical choice. People are using this remdesivir still, but this basically WHO tells that it is not a significant result. The final one is the vaccine that what we are going to see right here. We know the mRNA and these inactivated vaccines. Then we have the adenovirus vaccine, and then we have the protein vaccine from the Novavax. So just an overview of that, how this works. So you have the Moderna and the Pfizer biotech vaccines. So these are produced in the lipid nanoparticle and delivered. On the other hand, the Pfizer contains a modified nucleoside, which is a pseudouridine. And what it does, that it has been shown that if you have an RNA with a pseudouridine, even that is even although this is an mRNA plus stranded mRNA and the cell thinks that is an mRNA, there are some mechanism that the cell can destroy this by some of the TLR activation. But the pseudouridine actually suppresses that, and the pseudouridine is present as a modified nucleoside in the Pfizer vaccine. So once it is delivered, it will be translated by the ribosome, and it will produce the antigen. The same way, you can just have an exogenous protein antigen that can be delivered through the lipid nanoparticle, and that is also the same way. This is that Novavax delivers the protein vaccine in lipid nanoparticle. So you have the protein right here from the mRNA vaccine, or from here the exogenous protein delivered. Let's see that now what happens. So now, once these proteins come inside the cell and produced inside the cell, there is some of these proteins, they will have these secretory signals, so they are going to be secreted outside the cell because they do have these particular signal in there. They have the signal peptide. On the other hand, there is a class pathway where the proteasome will degrade some of these proteins and catch them. and they will show these on the surface of the cell. These are the muscle cell, basically, by the MHC class II complex. That will attract the cytotoxic or the CD8 T cells. This is one pathway, which is the cell-mediated immunity. But we are more interested into that some of these secretory pathway proteins will be loaded up to the MHC class II, and these MHC class II will now be displayed outside the cell, which will attract the CD4 T cells. These CD4 T cells will now wake up the B cells, which will from B cells will go into the plasma cells and secrete the antibody against that. So this is basically the two pathways of that by which the mRNA vaccine basically works. On the other hand, for the adenovirus vaccine, the adenovirus do contain this E1 protein, is a non-structural transcription factor. And if you delete that, the adenovirus can well tolerate that. So how it is produced, that adenovirus, there is a shuttle vector, and there's a transgene cassette where this spike protein is introduced, and you can just make an adenoviral recombinant vector. And this whole plasmid is basically transfected, and you can recover these recombinant viruses, which is expressing these spike protein on its surface. And this is basically your adenovirus vaccine, but they are replication deficient. They will not be able to replicate in the cells, so they are completely safe. There's another strategy where you can use a shuttle vector by a direct cloning of the transgene cassette, but basically it is the same process by which the plasmid-based recovery of the virus, you can do that from the cells, and these cells are produced in a large amount in large fermenter, and you just purify the virus from those. And this is basically the adenoviral strategy. The last thing that I want to just stop in this one, that the future of the mRNA vaccine is the amplifying mRNA vaccine, and probably what we have right here is the conventional mRNA that you can see that right here. You produce the antigen immunotherapy like this. The self-amplifying and the trans-amplifying vaccine will contain an alpha virus RNA-dependent RNA polymerase 1 through 4 subunits, which can amplify the gene of interest, or say the spike protein, to produce more of the mRNAs to do more antigenicity or elicit more antigenicity. And what we think that a lot of people are speculating, that as the many new strains are going to be developed, may come out, and if we need a strategy, all you need is a low amount of mRNA with the different sequence of these of these spike proteins, and that can be packaged by the self-amplifying vaccine without increasing the amount of RNA dosage. You still can get all the mixture of these variant type of mRNAs to produce all the variant spike protein for broader specificity. So these are in the pipeline. So I am so sorry about that, overshooting that, and I will take any questions. Thank you so much.
Yeah, it's a great talk from Kaus. Yes. If anyone has questions, please feel free to ask.
Hey, Kaus.
Yeah.
This is Dave Feliciano. Beautiful talk. It's wonderful. I learned a ton. Quick question about the mRNA delivery. One of the questions that I was wondering about in terms of the vaccine, is there any data that you've run across about specificity of cell uptake of the RNA inside of the RNA particles in regards to the vaccine? Is it just all cells that the particles are touching, or is there just something specific to immune cells?
Yeah, so basically what happens, it is my understanding is that, and anyone who knows or read about this, please make a correction, that it is an intramuscular, so mainly it's going into the muscle cell. And in that process, around that, during the injection process, there may be some which is not going inside the cell and floating in that tissue, in that connective tissue space. And in that connective tissue space, which is basically the layers in between these muscle fiber or the muscle cells, we will have some of the immune cells in there, and as well as some of these connective tissue cells like fibroblast and other cells. So any cell which will come in contact with these lipid particles are going to pick it up. And one of the things that this is a late suggestion that is also happening, that why usually people are getting really super fatigue and really pain after the vaccination. And there may be a reason for that, for the Pfizer mRNA, Pfizer as well as the Moderna vaccine, that the muscle cells, after taking up the mRNA, they are also presenting through the spike protein through the MHC1. And that will cause the cytotoxic T cells, and killing a fibroblast versus killing a long muscle cell is much more inflammatory and painful. And that's probably one of the reasons that that side effect is a little bit intense in that process. But there is an evidence that antigen presenting cells, dendritic cells, fibroblast, these can be in that tissue space. They can also pick up and present the antigen to the CD4 T cells. Emily asked, is in order to present the CD4 T cells, some of the translated protein at least has to get into the professional antigen producing cells like macrophage and dendritic cell. Yeah, exactly. So that's basically, that's the idea, that's what I'm saying, that there is in between the muscle layers, we have these connective tissue which will contain the professional antigen presenting cells like the macrophage as well as the dendritic cells. Absolutely, that is absolutely correct. Thanks, Emily.
Yeah, Peng, this is Matt Turnbull.
Yeah.
I'm curious if you've seen, in looking at the alpha versus beta coronaviruses, and some of the stuff that I've seen suggests that alphas may be more common in the United States and North America, for example, when you start getting into non-bat hosts. And regardless of that, the idea is that the alphas are much more prevalent than people have really wanted to admit.
Okay.
And so there hasn't been a lot of work looking at their host range and ubiquity and so forth. And I'm curious if you've seen in your readings, what is it about the tropism that's changing between the alphas and betas, and the likely prevalence of host jumping or host shifting between alphas versus betas, and the likelihood that, you know, we're on a 10-year cycle for beta coronavirus jumping right now.
Right,
zoonotic
exactly.
breaks, and where are we as far as alphas?
I mean, like, I really, it's a great question, Matt. I mean, like, I really did not look into the literature, or it is not known to me, so I don't know about that. But I think that's a great question to look into it. And yeah, it's very interesting if we just look at the beta coronavirus, 2002, 2012. And then 2020 is just like 8 to 10 years that they are jumping these, and it just has become a routine. And I will try to look up, and I will really appreciate if you come up with any kind of literature, let me know. But I am not sure that what is the difference in the tropism of these two, which is causing this from the beta, but not the 229E or the NL63, which are the alphas, which are there for forever. And these are usually the strains that people work in the lab as well.
Well, I think regarding those four previous, you know, now seasonal coronaviruses, there is some suggestion that, you know, there was a pandemic in the 1880s, and one of those may not have been influenza, but may have been coronavirus and been one of those four, that we've just seen a rapid adaptation to that in human populations and other virus. So it may be that they're occurring more frequently and more recently than we accept or we recognize. But
okay. No, it's a great idea. I will just try to look up. Thanks for that insight. But I never looked at that. But now we are just so focused with the beta. I should probably look at the evolution and those questions, but I didn't, frankly. there is so much ton there, and I'm just learning every day. Yeah, but thank you so much, Matt. Any other questions?
Okay, if we don't, we probably stop here. And the presentation will be recorded, and Thierry will edit that and send to everyone. I asked the cause, and he's very happy to share with you. So if you want to share with your student, I think this presentation contains a lot of good information. So if you want to share that with your student, you feel free to do that. And thanks to cause for his permission. And just send a friendly reminder, we do not have seminar next week. So next week, so for faculty meeting with dean using same time. So we will continue seminar 2 weeks later, so February 22nd. Yeah.
All right. Thanks so much.